Good afternoon, ladies and gents. My name is Mrs. Sillier, and today we are looking at the grade 11 formal test preparation for term 3. Now, I want you to ask your teachers if they haven't provided you with this yet for your ATPs, because it gives you a good indication exactly what you must study. Sometimes we get confused and then the children study unnecessary information and spend such a lot of time on certain things that they don't need to know. So just make sure exactly what you need to know. Okay, let's have a look. Let's look at the ATPs and then the different topics. All right, now on the screen here we have the annual teaching plan for 2021 for grade 11 term 3. And now what I want you to see is that they give us certain topics here. Here you can see core concepts, skills and values that we have to look at every week. But then sometimes in the activity, there is some additional information that you need to know that is not necessarily listed here. So today when I plan to assist you with the preparation for this test, I looked at the activities as well, and we hope that we covered all of them. It also depends on what your teacher is expecting from you additionally to know for next year, since we're getting closer to grade 12. So just make sure that you prepare yourself, rather know more than actually less. Okay, so let's start. Let's have a look. We are busy with the formal test preparation for grade 11 in term 3. And then again, when you get tired or when you want to eat something or get something to drink, obviously you can just pause this video, go and fetch it and then come back. Or like it is sometimes now, you lose interest or focus very quickly. So just to get yourself sorted, you can pause and come back and have a look later again. All right, now first of all, let's just have a look. What is the test format going to be? So the test format tells us exactly what is the skeleton, the recipe for your test. So we're going to have a section A and a section B. And as always in life sciences, ladies and gents, there will be a variety of short questions in section A. The normal multiple choice questions that you get. And then obviously the terminology that many of you feel very negative about, where they give you the definition and you just need to give us one word for the whole sentence. Very important to remember here that sometimes when that word has an abbreviation, please don't use abbreviations in the terminology, use the full word. And then sometimes when you're not exactly sure how to spell it, just try your best. If you spell it incorrectly and we are still able to make it out, maybe your teacher will give you the benefit or the doubt and then just ask you to drill the spelling later. Or maybe the spelling is too close to another life sciences term, and then obviously that's going to be a problem. You will not get the mark. Then the last one that we have there with the short questions is fit column A to B. They give you a statement and you have to choose between A only, B only, none, or both A and B. Okay, and then lastly, the data response question is where they will give you some information, a sketch, a diagram, a case study, and they will ask you questions about that. And that is the 20 marks for section A. Now, section B, there will be 2 questions, and they will be in total 50 marks. With the short questions of 20. So section B, we will have 15 and 15, and here's different options. Obviously there's going to be an investigation, a question where you will have an experiment where you have to explain to us, you know, what is the hypothesis or what is the investigative question, the dependent variable, the independent variable, what must stay constant, how do we get variety in this experiment, or actually how we get this experiment to be valid. And then also, how do we get reliability is the word I'm looking for. So always and forever, there's 2 ways that we do that, but we will discuss that a little bit later. Now, lastly, ladies and gents, there will be no essay question. So if you have old exam papers, it would be very good if you can practice the essay question, because normally it's divided into 2 sections, shorter sections, 5 marks or 7 or 8 marks, depending on the question. So then you can still practice that as if it's a shorter question of 5, 6, 7 or 8 marks. So you can still use the old exam papers. All right, so then this 50 marks, it will be 50 to 60 minutes depending on the amount of work that you have to read. And that is your test for this term, ladies and gents. Let's have a look what is the theory about. Okay, so now this is all about gas exchange. And before we can carry on with gas exchange, there's 3 terms we need to know exactly what these processes mean and what's the difference between them. And this, ladies and gents, you have to study. So the first one is this one, it's breathing. You do that every day. In other words, you have inhaling of oxygen and some other gases, and then exhaling of carbon dioxide, water vapor, and oxygen, a little bit of oxygen, etc. So breathing means the inhale of oxygen and the exhale of CO2, and that happens through your lungs. Okay, that's the breathing process. Now, when you breathe and the oxygen comes into your lungs, then we have gas exchange happening. Here you see a gentleman with the lungs here, and then we look at a closer section and you see the alveoli there, and then even closer they look at one of the alveoli, one lobe, and there we see like CO2 and oxygen, how they diffuse through this membrane. So now when the oxygen comes in, we're going to have gas exchange inside your lungs, and more specifically in the alveoli, where we have a process that we call diffusion, and that moves through a membrane. So what is gas exchange? The process where oxygen and carbon dioxide diffuse or move through a membrane or a surface. Okay, so now we have it in our blood system, and when it's in our blood, it's going to go to our cells, and in our cells, cellular respiration takes place, and that is of course to give us energy. So the glucose that you have already in your body, and then obviously in your cell, this and the oxygen are used to produce lots and lots of energy for you, and energy is in the form of ATP in your body. Now, as you can see here, although it's a little bit small now, this is the mitochondria or mitochondrium, and that is the powerhouse of the cell. Here's the cell. There's the different organelles in the cell that hopefully you will be able to identify, the nucleus. There in the blue section we have the cytoplasm, and then those small bean-like structures that you can see there, that is the mitochondria, and that is very, very important to help us to get energy. Now, when we use the energy, and we use the glucose and the oxygen to provide us with energy, the CO2 and the waste have to go back, and it goes back to the lungs, and their diffusion takes place again, and the CO2 is going out, it's going to be exhaled out of your body. So this whole process, these 3 things, they depend on one another. They are interchangeable. There's some relationship between them. So you need to know which one is which. Otherwise, when you use the term, you're going to be confused. Okay, that is plain and simple study work. Gas exchange. All right, now this is the lungs. The lungs that some of you unfortunately use for some other extracurricular activities like smoking, which is not really very good for you, ne? Now, for your lungs to actually work properly, it is built, it is functional, it's built in a specific way to do its job. Now, when we look at this picture, ladies and gents, there's now a human being. Like you can see, there's the nose and the mouth where you're going to inhale and going to breathe air in. We will have the pharynx there. I'll talk about that in the next slide. The larynx, which is your voice box. The trachea, which is a normal word that people sometimes use, is the windpipe, but you have to use the word trachea. Here we have the right lung. I'm going to make an R here, and here we have the left lung. Remember, when we look at pictures like this, it's like being face to somebody else. Your left hand side is not his left hand side. his lung will then be on the right-hand side when you are looking at this person face to face. Okay, so the lungs that you see here, we have 2 lungs, obviously. And then there's lots and lots of smaller structures inside the lungs that we call alveoli that's going to help to increase the surface area. So the first one, we have 2 lungs, and inside them there's smaller structures that helps to increase the surface area. So you can't see it that easily here, but you will be seeing it in the next slide. Here we have lobes, né? And every time when there's lots of lobes, like you know, when you have grapes, there's lots of lobes, then you can actually increase the surface area that way. Okay, so we need a large surface area. Just remember that the lungs you will find in your chest cavity. Sometimes we call it the thoracic cavity. And here, just underneath this thoracic cavity, we're going to have the abdominal cavity. Now, this structure that you see here is your diaphragm. The diaphragm is a breathing muscle. It works with your intercostal muscles that's in between your ribs here. It helps with the breathing process. And this diaphragm is actually a thin sheet-like muscle. Don't know if you guys can see the stripes here on the picture. It gives us an indication that this is now the diaphragm and that it is a muscle. And around the lungs, there's going to be a membrane that we call the pleura. And this membrane, there's 2 layers, and inside the 2 layers there will be a fluid, and that fluid helps with the prevention of friction. Okay, just to explain, your sternum will be here, your ribs will be here, and all of that is actually there to help to protect your organs that's inside. And the one that we are talking about today, that is actually the lungs. Okay, so like we said there, large surface area. We need a large surface area because the bigger the surface area, the more oxygen can come into your lungs and the more CO2 can be exhaled. And then the next thing that's very important, this surface area needs to be moist so that the CO2 and the O2 can move through. A thin surface area, very, very thin. And then the problem is with some people that has certain disease of the lungs, there's scar tissue. And the scar tissue then thickens that surface area. Then it becomes a lot more difficult to actually, you know, for the diffusion to take place. And then obviously for people like that's got asthma, that also decreases the surface area because these alveoli, the lobes, there's some sort of contraction taking place and then constriction, and then the person cannot get, it feels as if he can't get his chest open, his lungs won't take in oxygen. Okay, so all of these things can influence the gas exchange. The next one that is very important is the transport system, and the transport system that we have in our lungs obviously is the capillary system. Okay, there you can see blue sections and red sections. You must remember that the blue sections, that would be the CO2 rich blood, and the red sections would be the oxygen rich blood. We talk about oxygenated blood for the red and deoxygenated blood for the blue. All right. And then adequate ventilating system, in other words, for you to be able to breathe in and out. And then the protected surface area, like we talked about now, the ribs, and there's the pleura showing you that it's going around the lungs. All right. Now, we go down the throat here. Let's just look at this picture again. We start here with the nasal cavity. Here you breathe your nostrils, you breathe in air. And obviously inside your nose, you know there's a septum. It's separating the 2 openings. And it helps to actually channel the air so that it can go into your nose, where you will find mucus and you will find hair. And that is supposed to trap the dust and the particles that's not supposed to come in. And obviously to warm the air as well. And it will go through this section. And here you will see is the pharynx. Now that's in your throat. So when you go to the doctor and you are sick, sometimes the doctor will say you have pharyngitis, meaning that the infection is here in this area where you have the pharynx. when we go down, that big section that you see there actually is the tongue. And then here at the back, going behind your trachea, there will be the esophagus that's going to your digestive system. So when the food and the air comes in through your mouth, because you can breathe air in through your mouth as well, maybe when your nose is blocked when you are sick, then it comes in here, but then the food goes down the esophagus, it's supposed to, and the air goes here through your trachea. So to prevent the food from coming into your trachea, here we have a section that we call the epiglottis, that area. Now when food is swallowed, the glottis, that is like a cap-like structure there, and the glottis is closed by the epiglottis, and this prevents the food to go down the trachea, in other words, to prevent you from choking. All right, then the next one is the larynx, that's your voice box. And again, when you go to the doctor and you are sick, your voice would become raspy or hoarse, or you can't speak. Sometimes you just whisper, and that is because now you have laryngitis. Many times there's a lot of, maybe with sinus or sinus drip, it accumulates on the voice box, and then obviously it can't vibrate. And then that is why your voice becomes strange to you. The doctor will give you medicine to clear that up. All right. Now, as we go further down here, this pipe here that you can see, that is called the trachea. And there you can see there's cartilage, C-shaped rings. Obviously, the C shape is like this. Let me get another color, like a red that you can see. It is like this. So this is now actually to help the trachea to stay open so that the air can come in. And when you look, when we go from the trachea, here we're going to go to the bronchus, the bigger one, and then the bronchiole, which is the smaller one. And we have one going to the right lung and one going to the left lung. And I say again, remember in the picture, because you are looking at the picture, it's the other way around. And then obviously here it's going to become where we find the alveoli or alveoli. Okay, something else I want to say. The reason why the C-shaped rings are like this, and I want to say that the open section is towards the esophagus. So when you swallow something, and maybe sometimes, like what is your mom saying? Your eyes are bigger than your stomach, and you take big bites, then sometimes it feels as if it's hurting in your throat. But luckily the esophagus, when you have a big chunk in your throat, there's space, and it can sort of press into this section. So there's an opening. In case you take big bites, then there's space for it to move a little bit, use a little bit of space there. Again, the pleura here on the outside, and that's the different parts. Ladies and gents, please remember, you cannot explain or answer a question if you don't know the names and if you can't label. So make sure that you can label first, and then if you struggle to spell a word, then you just write it out yourself 10 times or 20 times until you can spell this and you know exactly how to do it. Okay, but very important, know the labels, because before you can answer questions, you need to know what it is called. All right, and here we carry on. This one you can see nicely. Let me just do this. Okay, here we have the voice box there, the larynx. Then we have the trachea, and there you can nicely see the C-shaped cartilaginous rings. And here you can also see, this is now, for instance, the bronchus or bronchus, né? So this is the one on the right hand side, this is the one on the left hand side. So one each, big one goes to the one lung, another big one goes to the other one. And then it divides into bronchi. And then the bronchi divides into these small ones, which is the bronchioles, né? And they are the smallest ones. But look here, you can't see any of the cartilaginous rings there. So we only find them in the bronchus and the bronchi. All right, so there's no cartilage here, and then obviously we're going to find here, we're going to find the alveoli, which is also called air sacs. I just included this little picture here to remind myself and you that sometimes in class your teacher is doing an experiment where we have a glass bowl, and inside the glass bowl this is now 2 balloons that is here, and then a sort of a rubber sheeting here at the bottom to illustrate, or they tried to show you how the breathing system works. So they're going to move this membrane, the diaphragm there, to change the pressure so that air can go in. In other words, the balloons can swell up and then deflate, inflate and deflate. To explain to you in class how the lungs work, just remember this glass container, obviously it's not like your lungs that can lift and this cavity can't increase. This cavity is going to stay the same. So it's actually just showing you how the 2 lungs increase and deflate, inflate, deflate, but remember there's certain disadvantages with this system. Just go and have a look at that in your book, maybe they ask you. Alright, so just make sure. Alright, now the next one, these lovely alveoli that we have here. Obviously, they can ask you, we love to ask you adaptations. Why is it looking like this? It is looking like this so that it can do its job. But before we look at that, let's just understand the picture. Okay, so this big structure that I see here is the bronchiole or bronchiole. It is inside the lungs now. We are inside the lungs. Ne? And there we go. This is the alveoli. So there's many of these bubbles is there, and it's increasing the surface area. Like it says here, it is lobed, meaning I have 1, 2, 3, 4. I have lots of them. It looks like Aero chocolates, ne? Bubbles, and they increase the surface area. This is how it looks on the outside. There we have the whole capillary network, the blood that is close to them. We'll talk about that now, and this is how it looks on the inside. Also, it shows you here the pulmonary artery. The moment you hear pulmonary, it means lungs. So it's coming and going to the lungs. Here you can see the arrow indicating that there's now blood coming in here. And then obviously the blood coming in here is CO2 rich because it's blue. The blood going out is oxygen rich because it is red. All right. So first we have what we call here the pulmonary artery, and then the pulmonary artery will become arterioles, which is smaller, and then lastly we will have this network of capillary. It looks like a net, ne? So it's a network of capillaries. They are the smallest, and that is what we have here. They took one of them and then illustrated a little further what is happening here, and this is very important for you. So one of these circles is actually one of those alveoli. Okay, it shows you the oxygen coming in, it shows you the carbon dioxide going out, because we inhale the oxygen and we exhale the carbon dioxide. Now again, those capillaries that you see there, here's only one of them. Actually, this picture was cut off a little. So there's only one. It shows here we have oxygen-rich blood, and then it goes back into the alveoli so that it can be exhaled. And here, these circles that you see here, this is the red blood cells, or sometimes we call them the red blood corpuscles. Okay, and I always say it looks as if they are dented a little bit like this. There's a dent, that's what that circle is showing you, but it's round, so it helps with the surface area. There's a side here and a side underneath and on top. That whole area can then obviously carry oxygen when it combines. I'll tell you now. But this is what it is showing here. And the size of this capillary is made in such a way that they're like soldiers that's standing upright like this, so that they can easily come and it's like a production line. It can get the oxygen, it can give back the CO2, and it can get the oxygen. And here is where our diffusion is taking place. Okay, so we started off by saying it's low because look at how many we have. It increases the surface area and it's taking up less space. Then the next thing, this is a single layer. It's a very thin layer, and it needs to be a single layer so that it can be thin, because then it's only a short distance for the CO2 and the O2 to be diffused over this membrane. All right. So another thing that happens here, we have rich supply of blood capillaries. We need a lot because we need the oxygen to come in and we need the CO2 to go out. So we need a huge or a richly supply of blood capillaries for the transport. Okay. And then they also tell us here there's a narrow diameter to allow every blood cell to come in a single file to receive and to give away the carbon dioxide and receive the oxygen. Then what happens here, the red is obviously oxygen-rich blood. So the red blood cells, there's a protein that we call hemoglobin. And then the oxygen, it combines with the hemoglobin. And that is the taxi for the oxygen to go to the rest of the body, the transport. Transport of oxygen happens with hemoglobin. It combines, it forms oxyhemoglobin. All right. And then the shape of the blood cells, the difficult name is, it's biconcave. Bi meaning two, meaning one on that side, one on this side. It's two sides, so it's biconcave. These things, study this, ladies and gents. This is how the alveolus is adapted to do its function. We like to ask you about adaptations. All right. Now, transport of the gases around the body. It's again the same picture. How do we study? We study with repetition, doing it again and again. Here you can see is the heart in the center. This picture obviously is not correct. It's just to explain what is happening here. There we have the one lung, there we have the other lung, and this capillary network is obviously around the alveoli that we can see here, the lobed structure. And then it just shows you here the gas exchange in the lungs. There we have the red blood cells in that single file. There we have the thin layer in the alveoli. This is the alveolus because it's just one, and it shows here the CO2 diffuses from the blood into the plasma, into the alveolus, and then it will be breathed out. And that one shows that the CO2 is coming in. It combines with hemoglobin, it goes into the red blood cell, and then it's distributed where it must be. It flows all the way to the body cells. Here's the body cells. You can see there. It shows us two different cells here. There's another one and there's another one. And this is now in the capillaries. What happens here is also diffusion, where the sugar and the oxygen goes into the cell for cellular respiration to give you energy. And then the waste and the CO2 is going back with the red blood cells to be exhaled. Okay, the CO2 that is. Now what you need to know is this. Hemoglobin, you find that in red blood corpuscles. Corpuscles is just another word for blood cells. So the oxygen comes from the alveolus and then it forms a combination oxy, referring to oxygen, hemoglobin to hemoglobin. Okay, and this bond is easily formed and easily broken because it needs to form easily to actually take the oxygen where it needs to be, and it needs to be easily broken so that the oxygen can go into your cell. If that bond was very strong, it would be difficult for the oxygen to go into your cells. Okay, and then the same with carbon dioxide, you see there. Carbon dioxide is inside the tissues. Now the hemoglobin that just carried the oxygen must now carry the carbon dioxide, and that combination is called carb-hemoglobin. So that's the form that carbon dioxide is carried in. We also see that most carbon dioxides, they react with the water to form an acid that we call the carbonic acid, which actually then makes it more acidic, the pH more acidic. That forms bicarbonate ions, and then some of the carbon dioxide is dissolved in the blood plasma. So when it comes to CO2 gas, let me do this so that you can see. CO2 gas will be transported one way as carb-hemoglobin, the second way bicarbonate ions, and the third way it dissolves in the blood plasma, the normal blood parts. Okay. Now, when it comes to the oxygen, here we have our oxygen. Oxygen, the first one is, it is transported in the form of oxyhemoglobin, and then also you get that in the blood plasma. Okay, so that's the two taxis, one for the oxygen and the other one for the carbon dioxide. All right, and then just some more information for you. When we breathe in and when we breathe out, what is the composition of the air? All right, so inspired, meaning the air is coming in. Expired, meaning the air is going out. So during rest, when we are not busy with an activity or when we are at rest, then we normally breathe about 15 times a minute. Now, when we have a look here, these are the different gases that you find in air: nitrogen, oxygen, carbon dioxide, and then obviously water vapor. If you don't believe it that you breathe out water vapor, you can just do that in front of a cold glass and you will see that immediately. Some of you must write love letters for one another on the classroom window using this, ne? So water vapor is also some, it's also coming out of your lungs, and there's water in the form of water vapor. All right, so now inspired air, air that you are breathing in. Here we have it. Inspired air we breathe in. Expired air we breathe out. Let's have a look. When we breathe in nitrogen, they say it's more or less 79%. When we breathe it out, it's 79%. Why? Because we don't use it in our body. The plants, they normally use the nitrogen. It just needs to be changed from nitrogen gas to nitrates in a form that the plant can use it. Then oxygen, here you can see 21% breathed in, 16% breathed out. Why? Because we use it in the process of cell respiration to actually do what? To give us energy. Okay, then CO2. There's not a lot of CO2 breathed in, but there's quite more, from 0.04 to 4. And why do we breathe a lot of that out? Because of the cellular respiration process. Let me just remind you, in case you have forgotten about this. Normal photosynthesis, ne? Normal photosynthesis is like CO2. Plants use CO2 plus water, and then it gives us C6H12O6 plus oxygen. That is what plants are giving us, because we want the oxygen. The sugar the plants use, they need chlorophyll as well, and they will need energy, and they will need enzymes. That is what that one is for. I don't have a space there. And obviously that is sunlight. So those things are needed for this to happen. This is now happening in the plant. Now we eat the plant to get the sugar. So cellular respiration, that's my abbreviation to save time. Cellular respiration, we use the sugar, the glucose, and we breathe in the oxygen to do what? To form CO2 and H2O. That's the water vapor, and that's the CO2 that you see, because we used it in the process of cell respiration. So we don't want it to accumulate in the body. It's toxic, and like I said previously, it's going to decrease the pH. It's going to be more acidic, and that is composition of inspired and expired air. All right, now the process of gas exchange, the process of breathing. Your teacher can ask you this. We sometimes give you pictures with arrows, and then you're supposed to identify, like for instance in this picture, we can see the arrow shows air is coming out. We can see the movement of the diaphragm, and we can also, this arrow is supposed to show you that the thoracic cavity, the ribs, and the chest cavity is now expanding. It's becoming bigger. If you don't know, people, you can just breathe in. When you breathe in, immediately you realize your chest cavity is now expanding. So what must you write when you talk about inhalation? So when we breathe air in, what is going to happen? the external intercostal muscles contract. Intercostal muscles is the muscles between your ribs. Ne? That's the one set of muscles that's going to contract. The other set of muscle, the diaphragm, remember that is here. The diaphragm is that muscle that we see there. The diaphragm is also going to contract. And when it contracts, it flattens. It looks as if it's pushing up your chest cavity. Pushing up your chest cavity so that the chest cavity can expand. What also happens is it's increasing, that is what that arrow means, it's increasing the total volume of your chest cavity and decreasing the pressure of your chest cavity. Alright, that is inhalation. Now what is exhalation? Here we see, look at the arrows. The arrow shows you air is coming out. This arrow shows you that your chest cavity is going to reduce. When you breathe out, your chest cavity reduces. And look at what is happening to the diaphragm. Ne? The diaphragm is pushing out the air. So the chest cavity is reducing and the diaphragm is arched there. It's staying there. Ne? It relaxes, it stays there so that you can get the air out. Alright, so thoracic cavity reduces, external intercostal muscles relax, the diaphragm relaxes, the total volume decreases, the pressure increases. Why is the pressure increasing? Because it's a smaller area and the volume, the amount inside is going to cause pressure because there's less space. Okay. Now, something I want you to notice, when this muscle contracts, the diaphragm contracts. When the intercostal muscle relaxes, the diaphragm relaxes. So please, ladies and gents, again, this is study work. We can give you pictures like this, or we can just plain and simple in table form ask you to compare inhalation and exhalation with one another. So that's a way that we can ask it. All right, next one. Effect of exercise. And you will say, ma'am, the effect of exercise is it's making me tired. Okay, it's too much. We are not used to exercise anymore because we may not do sport, social distancing, etc. But this is always a very interesting section for me. The effect of exercise on your breathing and your pulse rate. So now let's look at the breathing section first. Here I have Usain Bolt, so you will know about him. He's very, very, very fast. And now with exercise and with breathing, what is happening now? Now let's look at this first section there, what they tell us. They say in a sprint, you cannot breathe fast enough. So respiration takes place without oxygen. That respiration that's taking place in your cell to give you energy. Remember, these guys need a lot of energy over a short period of time. So there is anaerobic taking place. We need this anaerobic respiration, so it takes place without oxygen. Energy is still released, but much less. Lactic acid is made. This is a poisonous substance. It causes cramps. Oxygen needs to be put back into the lungs to remove the lactic acid that's going to be in your body. This is called the oxygen debt. And they say, watch sprinters after a race gasping for air to repay their debt. That section where they are finished, when they do this, it shows you exactly that they now have used a lot of energy to do that, to run fast. Okay. Now, here underneath, it's just to show you another picture, aerobic respiration when using oxygen. Okay. Respiration, cellular respiration, very important. Breathing, very important. If you need more oxygen, you need to breathe faster. And when you need more oxygen, many times we breathe deeper. That is, after practice, it serves no purpose to go and bend over so that you decrease the size of your chest cavity. You need to stand upright, actually to stretch your arms in the air so that you can use the whole surface area of your lungs. If you bend over, then you decrease this opportunity. So you need to stand upright to get enough air into your lungs to recover. The other one that we're going to look at is the pulse rate. In other words, when you are exercising, it increases your heart rate and then obviously your pulse rate. Now, where do we measure our pulse? There's two places where we can do this. Here in your neck, where that main artery is, and then obviously here just underneath your wrist, and you use your finger, not your thumb, to do that, 2 fingers, and then you can count what is your pulse rate, how many beats per minute. Okay, and this interesting graph shows us unfit person like myself and a fit person, how we struggle. Before exercise, you see there's the pulse rate 80, okay, and then when I exercise, my pulse rate increase here to almost 140 because I am unfit. And then when I'm finished with my exercise and I rest, it takes me a long time for my pulse rate to go back to more or less normal. Look there, there's 2 sections before I go back to normal. A person that is fit, first of all, this person's pulse rate will be lower. Your pulse rate is a good indication to tell us exactly how fit you are, and that is different for different people and different sports. Every person is different. You can't just take one size fits all and give everybody the same program. You have to look at the specific person. There, this fit person exercise, you see the pulse rate is never going so high because he practiced already. But the big thing I want you to see here is what is happening. While I am still trying to recover, the fit person recovered in half the amount of time. Look there, almost back to normal. There is stabilized recovery time. His recovery time is much less than my recovery time. My recovery time is that whole area there because I am unfit. Okay, so that influences also your breathing, it influences your heart rate, all of those things. Very important to remember. Another interesting topic, exercise and altitude. What is altitude? Altitude is where exactly above sea level you are. In this picture I have a mountain, and then it shows you increase in altitude. The higher I go, higher the altitude, the lower the pressure and less oxygen. Look at that section. Those circles that you see there are supposed to represent the amount of oxygen. When I go at sea level or lower down, going down the mountain, it's a lower altitude, lower height above sea level, greater pressure, because the pressure that is now on top of me, I just want to show you this, maybe you can't see it that clearly there. If I'm now standing here, I'm Parawena there, all of that pressure is on top of me. When I'm standing here, the only pressure that I have on top of me because I am higher is this amount of pressure. So the higher I go, the lower the pressure, the less oxygen. More towards sea level, this is now sea level. When I'm here, the altitude is lower, there's more oxygen, but the pressure is more. Very important to remember, if I'm now underneath the water, that is also important. You can't see my little fish here. If I'm underneath the water, it's the added pressure that is now on top of the sea level and underneath the sea level. And if I'm diving here, it's added pressure. So when the divers normally come back, they have to stabilize or equalize at different levels. They can't just come back. Then obviously that's going to have an influence on them and make them sick. Okay, that's the one thing. The other thing that I want you to know is people use this to exercise, to improve their performance. Okay, so when they train, let's say they are running, when they train at sea level, there's lots of oxygen. We normally say we run easier, if running can be easy whatsoever. But we run easier at sea level because there's lots of oxygen, we can make lots of energy. Okay, but when we go up higher, there's less oxygen to go with our red blood cells. So now your body is very clever, it's compensating, it's making more red blood cells, because if there's more red blood cells, you can carry more oxygen, okay? So now your exercise, they say usually it must be a minimum of 4 weeks for your body to adapt. Now you exercise here, you have loads and loads of red blood cells. You come back to sea level. When you race here, I don't want to say you have double the amount of red blood cells, but you have much more. So now you can carry more oxygen and you will even run easier than what you did before. Okay. So this will not last forever, but this will have an effect for some time. So that is also a tool that people use to help them to enhance their performance, exercise and altitude. All right. Now, very, very important, ladies and gents. Your teachers may put a specific emphasis on this because this is also part of grade 12 work. So you are going to see this again. All right, now homeostatic control of breathing. That sounds very difficult. All that it means is we always want homeostasis or homeostasis in your body. That means that your body must be in balance. You cannot have too much or too little. It must be just right, like in the story with Goldilocks and the 3 bears. The porridge is too cold, the porridge is too hot, the porridge it's just right. So all the different things in your body, I'm talking about glucose levels, thyroxine levels, oxygen levels, carbon dioxide levels, water levels, salt levels, all of that must be in homeostasis, in control. It must be what we say here at a normal level for your body to actually work much better. So this is called the homeostatic control of breathing, to keep the CO2 levels in your body normal, because CO2 can be toxic when it accumulates. So what happens? Here's the scenario. Physical activity increase. When you exercise, you have to breathe more because you think you get tired, but your body needs more oxygen to make more energy. Cellular respiration increase. Okay, so that increase. The moment I have more cellular respiration, because CO2 is a product, I have more CO2. So CO2 is produced, it's increasing. Then inside your brain, there's a very clever section, the medulla oblongata. Now the medulla oblongata is the detector, not the detective. It's now realizing that there's too much CO2 in the blood. We need to make a plan. So it's sending an impulse to 2 places. The first place it's sending an impulse to is the heart. The second place is to your lungs. So what must the heart now do? Let's have a look. The heart rate increases because if the heart pumps the blood faster, the CO2 will get out of the lungs faster. It will get to the lungs faster and we can breathe it out. And so the CO2 levels drop. When your breathing increases, the depth of your breathing increase and the rate increase, rate meaning how fast, ne? So you are exhaling CO2 more rapidly, faster, and then the CO2 level drops. I don't really like the word drop. It decreases. And it's not just decreasing, it's decreasing back to normal. That's the words that you must use. And that is homeostatic control of breathing. Like I said, important for grade 12. So I guess it's one of the questions that your teachers would really like to ask you. Okay. Now, that was now breathing. Now we get to excretion. All right. Now excretion in humans. We have certain organs that is now excreting certain substances. As you can see here, we were talking about the lungs. The lungs get O2 in and it gives CO2 off, so to speak. Inhale, exhale. CO2 is a waste product, so it must go out. It's excretion. Then now we're going to look at the kidneys. There's the 2 kidneys, the bean-shaped organs. They're also not at the same height. If you can see here, there's your pelvis. There is your bones, so it gives you a good idea where you can find your kidneys. There we have the ureter, that's going to your bladder, and there's the urethra. So what is excreted here? Urine. obviously. So that is part of the excretion. The alimentary canal, there's the mouth, then we go, there's the liver, there's the stomach, there's the pancreas, we have the large intestine, the small intestine, the rectum, and the anus, and the appendix, which is a vestigial organ. So that you already know because you studied this ever since primary school. Okay, so what is excreted here? Feces, obviously, all of the fiber and other substances that your body no longer needs, it needs to come out. Okay, then skin. Skin is also important for grade 12. Look at this. There's all the hairs sticking out. We have the different layers of the skin. There I can see that's the sweat gland with the sweat duct, and there's the sweat pore. You can see holes there where the sweat is coming out if you are sweating, not if, when you are sweating. You are trying to regulate your body's temperature to cool down, okay? This is part of the hair that shows, it goes out of your skin. It shows here. Then there's adipose tissue, which is fatty tissue. And again, I have blood vessels. There I have the CO2 rich. There I have the red ones is the oxygen rich arteries and veins. All right. I will also have some nerve cells here in between, nerve tissue, because they need to get the information. They are receiving information. And then lastly, the one here is the liver. The liver has many functions, but the big one here is it is now forming, or it's actually changing excess amino acids. They break it down to form urea. We call it deamination. Amino acids that we are not using, that's not absorbed in the body. There's the liver. So these organs, they all have to do with excretion, where we have to get rid of some type of waste that's in the body because it's bad for us when it accumulates. All right. Now, again, before we can answer questions, before we can understand anything, we need to be able to label this. Okay, these are the things that you must be able to identify. Now, let's look at the picture. There's a body. All right. There's the kidneys. They are bean-shaped. All right, they have, it looks like a cap there. That's a gland. We call this the adrenal gland. The adrenal gland secretes 2 hormones. That's very important also in grade 12. Adrenaline, which you will know about, and that makes you do things faster. Your heart beats faster. The fight or flight reaction. Or aldosterone, that has to do with salt, which is also relevant here. Again, I showed you the right kidney, the left kidney, because we're looking at the picture. So it's opposite sides. There we have the renal artery that is carrying blood waste to the kidney, and we see oxygen rich. There we have the renal vein, and it's taking waste and CO2 away back. This is the left kidney, that is the right kidney. Okay, here we have your bladder. And this duct pipeline that we have here is the ureter. It's bringing the urine to the bladder, and this bladder can more or less plus minus carry 600 ml or keep it there temporarily before it's excreted in men and women through the urethra. Okay, urethra is going to excrete the urine. If some of you may think about this, when a woman is pregnant, the fetus normally presses on this bladder, so there's not a lot of space. That's why they always say women feel that they want to urinate frequently because of the pressure that they can feel on their bladder. So the kidneys, ureter, bladder, urethra with their functions. I'm going to look at that now. Structure. All right, make sure you know that. The kidney, now we look at the kidney up close and personal. We take one of them, cut them in half, and this is how they look inside. Very important again. Let's have a look. The first thing I want you to know is that on the outside we have the capsule. The capsule is a thin membrane that you get all around the kidney. And there just inside the kidney all the way around we get the cortex. When it's called renal, renal refers to kidney. You will see everything here is renal. Like when you look at the lungs, it's pulmonary. When we look at the kidney, it will be renal. Okay, so here on the outside, there's a whole section there on the outside is called the cortex. And then here on the inside, the medulla, the middle section is the medulla. We have it there. Then another thing that's very important, you can see, I'll use another color. You can see these structures here. And when you look at their shape, they say it's cone-shaped. Yes, cone-shaped, like an ice cream cone, like that. Ne? So it's cone-shaped. They are called pyramids, like the Egyptian pyramids, all right? They are called this because of their shape. We say their shape can be conical or cone-shaped. You can see that easily, all right? Now then the next thing that's important here, these structures where the pyramid goes into this bigger area here, this bigger area underneath those renal veins and renal nerve and renal arteries, that section that you see, that cream section there, is called the pelvis. So the major and the minor calyx, it just opens up here in the pelvis. And there the pelvis collects all the fluid, the urine, and then it goes down the urethra to the bladder. And this is where the urine goes. Okay, where do we find the kidneys? The kidneys is just below the diaphragm. So again, you can see all the blood vessels here, the veins and the arteries. They are very important to help with the function of the kidney because the function is very, very specific. You can't, if the kidneys are not working, you're going to become very sick. And then maybe later, if dialysis is not working, in other words, a machine that's doing the job of the kidney, then they have to get somebody to donate you a kidney. And you can become very sick before that happens. All right. Now, I have this huge summary here. I normally do it like this for myself. I have the picture and then I have the labels and then I summarize all the important things on one page, and then it's not so intimidating when there is 6 pages in front of me to study. All right, now I want to go back to this one first. So the function of the kidney will be filtration, reabsorption, and excretion. But inside the kidney, there's millions of functional units. And these functional units are called a nephron. And this is what the nephron looks like. So the nephron is this section in the kidney that's doing the hard work. And there's not just one, there's millions of them. And look at this picture, you can see where you can find them. Okay, you find them there and they are doing the following. So let's just have a look at the different parts first. So here you can see, excuse me, the renal artery and the renal vein coming in. There we have a structure looking like this. Okay, that structure I want to use, let me use the green. We have that there. There it is, maybe you can see that better. That green structure, there it looks like a head. And then this long tail section that goes into this collecting tube. So there's sort of a cup there, and then there's a long tail. Okay, so now we need to name these things. Now, first of all, this section here inside, that is blood vessels. That is a network of blood vessels. That is called the glomerulus. And that is where we find the first network of blood vessels. Now, coming into that network, we have the afferent arteriole. If you're scared you're going to forget which one comes in first, A is the beginning of the alphabet. So the afferent arteriole comes into the glomerulus network, and then the one that goes out there is the efferent arteriole. And this one is wider and that one is narrower because it needs the blood to be under pressure for all the processes. The substances must be forced through the pores like I have here. So that's the first one. The first adaptation. The afferent arteriole is wider than the efferent arteriole. You can't really see it here in the picture, but that is a fact. And it is wider so that it can be under pressure. The content can be under pressure so that the substances will be forced through the pores that we're going to get here. Okay, so that network is the glomerulus, and then this capsule, this bucket, this cup there, it looks like it's doing this, né? This capsule is called Bowman's capsule. And Bowman's capsule is now close to the network of arteries and veins, because the closer it is, the easier the filtration is going to take place. Because that's the first function that is happening here, filtration. All of the substances that can be used again must filtrate through, and it's only the smaller substances, because here, inside this structure, there's going to be small pores. And these small pores are going to be between specialized cells that we call podocytes. And they are small, these pores, because large molecules cannot pass here, like blood corpuscles and proteins. They are too big. They cannot pass through this small pore, so they stay behind. Only the small substances can pass. And like I said now, it's cup-shaped, Bowman's capsule, so that there's closer contact for this to happen. Then another thing that you should remember by now, there must be just a single layer of cell. We need a thin surface so that filtration can take place. There we have it. Thin surface so that filtration can take place. All right, here we go. Next one. That now I said to you was the first capillary network. You see it looks like that together like this. Ne? Now the next network, the second capillary network is this one, this larger one. That one is in a small area. This one covers a larger section. It circles all around these tubes that we have here, or tubules that we have here. Okay, and because it is there, it's covering a large surface area. That is very important. It's a large surface area. Then something else. This tube, it comes from here, and there it circles all the way to here. There's three different labels that you must know. The first one, the proximal convoluted tubuli. Proximal meaning it's closer to the head. It's closer to the proximity. It's closer to Bowman's capsule. It's this first section. Proximal and convoluted means it's folded like that. It's bending and making, it looks like you're driving on a mountain pass. All right. Proximal convoluted tubuli is the first one. So when I go through the loop of Henle, that looks like this to help with the whole process, then I get the distal convoluted tubule. Because it's further, it's in a distance. It comes later. It takes longer to get there, ne? Convoluted tubuli. And then that goes, and there's the collecting duct. And you can see, obviously, these branches that they are showing us here is other convoluted or distal convoluted tubules that comes in here that brings urine. And this is now obviously going to go to through the different parts of the kidney to go there into the pelvis and to be accumulated there in the ureter. All right, now have a look here, the green section that I've tried to highlight here for you. It is very long. It's not just going straight. That would have been a very short distance, but now it's long and is curled up and coiled like that. Ne? That is to make sure that there's a larger surface area so that there's more time for substances to be reabsorbed. Ne? Here we have it. It's longer tubes so that there's more time, opportunity for reabsorption. Okay. then you can't see it here. But the next thing that is very important, energy. Nothing can happen without energy. I assume your energy is also now used up by now. Maybe it's time to stop the video and get something nice to eat or drink so that you can just get the energy up there where it belongs again. So, what or which part? Mitochondrions. There will be many mitochondrions there to help with the provision of energy for this to take place. Then another thing that is important, inside these tubules, renal tubules, meaning the proximal convoluted tubule, the distal convoluted tubule, and the collecting tubule. They are all renal tubules because renal refer to the kidney. Those are the tubes we get in the kidney. And there are microvilli inside. In other words, inside the surface area looks like that. And when it looks like that, these finger-like structures, that increases the surface area. Okay, and then the last thing, if I go back here, this is now the kidney, né? The medulla is that middle area, the medulla. Medulla sounds to me like middle, né? There's where we have the sodium pump. Sodium is salt in the medulla. When there's a high, that increase of concentration of salt, so that there's reabsorption of water, so that that can take place. So when I look at this, this is how the nephron looks. It's inside the kidney. This one is doing the job for the filtration, for the reabsorption, and for the collecting of urine that is going to go to the bladder. Very, very important page. Know your adaptations. We had the adaptations of the lungs. We had the adaptations of the alveoli. Now this is the adaptations of the nephron. Its function and structure, always. It's built this way to do a job. So you always can explain why it looks like that. That is your job to explain to us to get marks. All right. Another picture just showing the same thing. If science is your thing, there we have some chemicals showing you the scientific language. We have salts there, sodium, potassium, calcium, magnesium, chloride. There we have bicarbonate ions. We have water. So it shows you here we have a Bowman's head. There filtration is taking place. What is filtrating? The useful substances is water, salts, glucose, amino acids, fatty acids, and glycerol. And if you know that that's the building blocks, fatty acids and glycerol is the building blocks of lipids, of fats. Amino acids is the building blocks of proteins. Glucose, building blocks of starch and sugar. So it's the smallest parts, it's the building blocks. Your body absorbs, digests food and take them back or change them to their building blocks. The smaller parts, they are useful. Filtration. The waste is urea and uric acid, and there can also be blood substances there, blood corpuscles and blood proteins, but they can't be filtrated, they can't go through, they are too big. There we have the proximal, in proximity, close to the Bowman's capsule, proximal convoluted tubule. There we have the loop of Henle. There we have the distant. So proximal is before the loop of Henle. Distal, distance, its inner distance is the one after the loop of Henle. And this whole section is where reabsorption takes place. Reabsorption of substances that we need. Reabsorption of water. Reabsorption of salts, of glucose, that is if we need it. Ne? Reabsorption of those things, or if we have too much of it, where it just, the permeability of the membranes decrease so that all of these things can just go out with the urine. If there's glucose in your urine, obviously it's an indication that you may have diabetes mellitus. Okay, this is the collecting tube. This is where excretion is taking place. There's the 1, 2, 3 main functions of the nephron, the functional unit of the kidney. Filtration is taking place there. Reabsorption is taking place in this whole area, and that section is excretion. What is excreted? Ammonia, creatinine or creatinine, potassium, hydrogen, sodium, bicarbonate ions, certain drugs like penicillin. If your body cannot use the vitamins that you are taking in, vitamin pills, your body can only absorb a certain amount. If it's not used, you just urinate all of that out. So that will be in your urine. All right, structure and function. Now again, here you see that difficult thing. Homeostatic control of water. NB for grade 12. So I think your teachers are going to ask this in grade 11 as well. What is this? Osmoregulation. Osmo is the regulation of the water potential of the blood. And it's very important to have homeostasis in the human, the correct balance of water. You can't have too much or too little. We just have we need the correct balance of water. When the water level in the blood is normal, we have homeostasis. Now this, what we have here in grade 12, we will tell you, it's called negative feedback. For now, you can talk about homeostatic control of water. All right? Now what happens when you drink a lot of water or you don't urinate a lot, the water levels is going to rise. It's going to rise above normal. When the water levels rise, the hypothalamus will get the information. And the hypothalamus will let the pituitary gland know to secrete a hormone that we call ADH, which is an anti-diuretic hormone. Diuretic is something that makes you dehydrate because you urinate a lot. So now we want less ADH. So the kidneys reabsorb less water. In other words, here in the tubes, less reabsorption takes place. The membranes will not be as permeable. So less water will be reabsorbed to go out through the urine. It stays in the tubes and then it goes into, if I can go back, it goes into this blood network. It's reabsorbed by the body to be used again because you don't have enough blood, and then the water level goes back to normal. Now, when there's not enough water in the body, again the hypothalamus will detect the high concentration of solutes, because remember, if there's now less water, there's a higher concentration of minerals and salts. The hypothalamus will detect that, will send a message to the pituitary gland to secrete more anti-diuretic hormone, and on the other hand, it's also going to create a feeling of thirst, so that you, in case you don't have enough water to reabsorb, that you can drink some more water. And then again, what happens here now in these tubes, there will be more permeability of the membranes, so that more water can be reabsorbed into the network and go back into your body. And then less water will go out through urine. And then what happens? The water level goes back to normal. Very important, people. I want you to mention the hormones involved. I want you to mention the parts in the brain involved. I want you to mention the words, instead of drops, to say increase or decrease, whatever is relevant there. And then it goes back to normal. Don't just say it decrease. It decreases back to what? To normal. Okay, there's that one. Homeostatic control of salt. Now salt dissolves in the water. So the same story. Okay, we want a normal sodium level. I say again, this is important for grade 12, so teachers may ask you this. You want a normal sodium level in your blood. So let's say this sodium level now increase above normal. Then the adrenal gland will secrete less aldosterone, so that less sodium will be reabsorbed, and then more sodium is lost in the urine. It goes out of the body, and then the salt levels in the blood decrease back to normal. If you forget which hormone is involved here, maybe this will help. Aldosterone, there's an S for salt. Okay. The same on this side. When the sodium levels decrease below normal, the adrenal gland is going to secrete more aldosterone so that more sodium will be reabsorbed, less will go out through the urine, and because more is reabsorbed here, the salt levels will increase back to normal. Okay, homeostatic control of salt. Please study this. This is study work. All right. Okay, so that is now the lungs, the kidney, all of those things. Now we're going to look at population ecology. Here, I think this is even easier because it's just study work and then they can give you graphs. So you just have to read. Population ecology, we start with species. What is a species? Here we see wolves. They are all the same species. They're in the snow, pack of wolves. Ne? So we will say it is a group of organisms and they have the same characteristics or traits. They are able to interbreed, to mix with one another, and when they interbreed, they will have fertile offspring. So all of these things that I have highlighted, that is your definition. We can ask it in terminology, we can ask it in normal questions. Those are the things that you must mention to explain to us what is a species. All right. Next one. What is a population? Now, we get species inside a population. So again, when you look here, it's the same thing. Let me just move this a little bit. It is a group of organisms that they have the same characteristics, okay? They are able to interbreed and they produce fertile offspring. So if you know the definition for species, you're almost there for the definition for population. The only thing that we need to add here is that they are in the same area. They are in the same habitat at the same time, obviously, otherwise they won't be able to interbreed. Okay, so population, a group of organisms with the same characteristics or traits that are in the same habitat, they are able to interbreed, and when they interbreed, they have fertile offspring. So there I just have a population of pine trees, okay, just to give you some variety. Now we go from population to community. The community. What is the community? It is a group of different populations living in the same habitat. Okay, different populations. Populations are made up out of species, so there's going to be different species. When you look here, it helps. This is the individual. Individual means this is the species. They have the same characteristics. They are able to interbreed, and when they are interbreeding, they have fertile offspring. What is a population? Group of species that are in the same habitat. What is a community? It's a group of different populations. Here I have a hare. There's an owl. Can't really see what is that. It looks like a moose, that one. So and then obviously the plant life that makes in a community. The community will make an ecosystem. The different ecosystems will be biomes, and we find the biomes in the biosphere. And remember, the biosphere is made up out of grade 10 work. What was it again? Lithosphere, atmosphere, and hydrosphere. Okay, ecology. Just back to the basics. You have to know the definitions. Okay, now the new things that is important here. Population size. Now, ladies and gents, it is very important to know what is the population size, because if there's too many people, there will not be enough resources. Then they're using all the resources, and then people can die because there's not enough food, or organisms can die. Now, two things first that's going to influence population size, that would be immigration. Now, immigration is the one way inward. Immigration, they are coming in. Here I have a fish population. The population is going to increase because some of the fish is coming in. They are immigrating. When there's an outward movement, when they go away, when they move away, we call it immigration, exit, coming in, going out. So this one, they're coming in, and this one, it's exit, they're going out. So when they come in, they're going to increase the population. When they go out, they're going to decrease the population. Okay, population size influenced by immigration and emigration. Next one. Population size is influenced by mortality and natality. Mortality, mortuary mortality, when we have death, okay? And then natality, reproduction, and the natal unit, natality meaning birth, these two, they must be swapped around. So mortality is decreasing the population size. When people die, there's less people. And natality is increasing the population size. There we go. So there's less people. Mortality, death, decreasing the population size. Natality, reproduction, more people, increase the population. There we see it. There we have birds. Again, they die because they get eaten or they become sick. It decreases the population. Okay, when there's small birds being born, it increases the population size. Remember mortality, natality. Okay, now this one, very, very important. Fluctuations and limiting factors. There's things that is influencing the amount of animals, organisms, people on a specific area. Okay, now the carrying capacity is determined by the resources available. If there's enough water, if there's enough food, there will be a lot of organisms there. The less food we have, or the more difficult it is to survive in that environment, the less organisms will be there. Only those that's adapted to survive in that area. Okay, if the resources, if it's in a short supply, it's a limiting factor. It is influencing this, né? It's lowering the carrying capacity. Now, carrying, when you carry something, look at this picture, you can only carry a certain amount of individuals, then it becomes too much. What this picture is showing us is that the earth can only look after a certain amount of people, then we will run out of resources. So, carrying capacity is, there comes a time when a habitat cannot support any further increase in numbers. So that is the number that a certain area can sustain. Okay, so if a certain area cannot sustain the population anymore, there's limits, like there's not enough water, there's not enough food, and that will be a limiting factor. Here you can see, trying to indicate carrying capacity. All right, now another one. Here we can give you a case study and ask you questions about the case study. Here we have some elephants in nature, okay? And the term that we need to look at is culling. Now, culling is killing of animals, né? But now culling is a legal killing of elephants in this case, or animals. When the population size becomes too high, then it starts to influence the environment. Like elephants, for instance, they eat from trees, the marula tree, for instance, and then because they are so big and they use up a lot of space, they may damage the environment. So if there's too many of them in a certain area, it can become a problem. Now, culling should be the last solution to the problem. Normally what they do, they say, for instance, in the Kruger National Park, they will say that the elephants, they are now a victim of their own success because they survive so well in the area that their numbers increase, and it increases in such a way that the environment is not, the carrying capacity is now not enough. Okay, there's more than what the area can handle, and then they destroy the environment as well. So what people will do, or specialists will do, is they will first of all try to translocate a family of elephants to take them somewhere else. Okay, and elephants is social animals, so you can't just take them individually. It's not good. They will experience a lot of stress. Even the movement will be stressful to them. And then another thing that they try to do is contraception, but that is also, anything that you do can be stressful to the animal. And then obviously it's also expensive. Okay. So we need to make sure that we look at the whole picture, the carrying capacity and what is good for the elephants, what is good for the environment. So culling only happens as a last resort when all of the other things that they have tried is not working. The legal killing to reduce the numbers of animals to prevent the effects that they have on the environment. All right. Now, how do we know how many animals do we have in a specific area? We have two ways of determining the population size. Now, the first one is the mark recapture method. Now you can have a look here. What it basically means is that you trap a few animals, you put a mark on the animals where you can easily see it, but you must be very careful that you don't use a mark that can be harmful to the animal or make the animal more vulnerable to predators. So nothing must actually happen to the animal because it is now marked. It's only for you to know. It mustn't be a chemical that's going to harm the animal. Ne? And then what you do is we use this formula, for instance. And the number in the first sample is multiplied with the number in the second sample and the number in the second sample previously marked. So what you're going to do is in a trap, for instance, you will catch animals and you will mark them. Then you will let them go. But you will give enough time now for them to mix again with other animals. And, you can't just do it the next day. You must give time. Otherwise, your results will not be, valid. So when after some time, now you catch animals again. Like, for instance, in this example, 10 animals were trapped, they were marked, and then they were released. 2 days later, 20 animals were trapped. Of these, 5 were found to be marked. So I start with number in first sample, that is 10, is multiplied with the number in the second sample, that is 20, divided by the number that is marked. So there we have a 10 times 20 divided by 5, it gives you 40. So that's our estimation. We are now making an educated guess. So this is the first way that we can actually determine a population size, and it's called the mark recapture, or they say capture recapture, that the words actually tell you what is happening. When we ask this in your test, we must give you the formula. Okay, so you must just be able to know how to apply the formula. Okay, then the next method that we use is simple sampling. Simple, not meaning stupid, meaning it's basic, it's not difficult. Here you see a learner that's writing information on a piece of paper. That structure that you see there, we call a quadrat. So there's the huge field. I don't know which type of sports field this is or just a part of the school grounds. But he's now counting the amount of dandelions that is on the school field. And the dandelions can give us a flower like that. And some of you blow the dandelion and wish for something good. And that is what I'm talking about when I talk about a dandelion. So now we use this formula. We say the total number in the population will be the number in the sample. There I can see 3, for instance. So what is happening is you're counting how many of them you have in this sample, and then you know what is the size of this quadrat. Okay, so the number in the sample multiplied with the habitat size, in other words, the size of this whole field, and then the sample size is the size of this quadrat, and then the answer that I get can give me an indication. So let's have a look. They say in a football field of an area of 5,000 square meters, 3 dandelions were found. You see 3. In a 10 square meter plot chosen randomly, estimate the total population of dandelion plants on the football field. Yes, that is not 10 meters, but that's just to give you an indication of how it works. So the number in the sample will be 3. It's multiplied with the habitat size. That will be the 5,000 square meters. And then it's divided by my sample. My sample is 10 square meters, and that will be 1,500. So I estimate, if this is the amount of dandelions in the small area, I estimate, educated guess, that there will be in that whole area 1,500. It's not always physically possible to go and count every single thing, especially with organisms that's not sessile. What is sessile? Meaning they are not sitting still or standing still, they move. It's easier to count something when they are in one place the whole time. So it just makes this easier. Mark recapture and simple sampling. All right, we're almost there, ladies and gents. Now, we are now estimating the numbers. Now we're looking at the growth of the population. Now there's two graphs that you must study for the test. The one is the geometric and the other one is the logistic. Now we normally just talk about the J-shape curve and the S-shape, referring to the shape of the curve. Ne? And here I summarized for you what you need to know. Okay, on the independent variable we have time. On the dependent variable, we have the population size. So you can see over time, the population increase. Now, this first section that we have here, we call the lag phase. There's little or no growth or slow growth because the animals are getting accustomed to the environment. Where's the food? Where's the water? Where can they be safe? Where's the predators, etc. And then there's exponential growth, and it grow and grow and grow until or while there's no limiting factors. And at some stage, you can see now it stops, the growth stops or stabilize. That's the geometric growth form. Logistic shows us that you reach the carrying capacity. When you reach the carrying capacity, it means that we now can't have more animals. The moment there's too many of them, we will run out of food, we will run out of water, and if they don't move away, which is going to decrease the numbers, they will die, which is also going to decrease the numbers. So this curve again, we start with the lag phase here. What happens is that there's little or no growth because the organisms are getting used to the environment. Where are they safe? Where can they sleep? Where do they get food? Where do they get water? Who's the predators in the area? Then we have the geometric growth form where there's lots of growth, and then at some stage it will balance out. There will be an equilibrium, and then if the graph goes down like that, it means that they are dying. That's the two growth forms here. Yeah, I just made a summary for you again that you can have a look. The population will increase because members of the population will be able to produce offspring, and after some time those offspring will produce their own. And this will happen under ideal conditions with unlimited resources. This population can grow exponentially. Okay, the logistic growth form or the S-curve, they say sometimes the population grows slower for several reasons because of the birth rate or the immigration rate. Density-dependent limiting factors will be competition for food, for space, for water, for light, for interbreeding. They can become sick because of parasites and diseases, and then obviously also because of predators that can kill them. So that is now what you need to know. That's the theory, ladies and gents. So I just quickly want to show you a few other things that you have to practice. Exam paper and investigative questions. Just again, I know you will be sick and tired, when you hear investigations. I'm sure your teachers are practicing this with you many times. What do you need to know when it comes to investigation? What is the independent variable? Okay, when I look on a graph, let's do it there, I will find the independent variable on the x-axis, this one. The factor that the investigator chooses to change or manipulate. In other words, the one that I can control, I normally say, that's the independent variable. It starts with an I. Dependent variable is this one. It is influenced by the independent variable. As I change this one, it will have an effect. So that is, the dependent variable is the one that can be measured or observed. That's the result of the experiment. And then there's some of them that must be fixed, that must stay the same amount of water, the same type of soil, the same amount of sunlight, anything that must be kept constant in order for this variable to be manipulated. Okay, then you must be able to write a hypothesis. In the hypothesis, we will have a relationship between the two variables. If the time increases, the amount of glucose that will be produced increases. There will always be a relationship. When the one increases, the other one decreases, or when the one increases, the other one will increase. And then very important, it is not a question, it is a statement, and it must be testable. All right, and very important here, ladies and gents, when it counts 2 marks, you don't get half a mark because you have the first part correct. It's either correct or not. So it's 0 or 2 marks. There's no in between. Remember that. All right. Something else that I just want to emphasize is reliability and validity. Reliability always and forever. What do we say when they ask you how do we make an experiment reliable? You repeat the experiment several times or more than once, and you increase the sample size. Okay, nowadays I see they do it the other way around. They give you the answer and then they ask you to repeat the experiment or increase the sample size. They, for instance, will say, what is the influence on the experiment when I measure the pupil diameter of 1,000 women instead of just 100 women? Then you can say it improves the reliability of the experiment when you increase the sample size. Okay. Then the other one that's causing problems, that is validity. How do we make an experiment valid? Okay. That is the method, the way that the experiment is now handled. What must stay the same, okay, in order for us to be able to test the independent variable, or to manage to control, to manipulate the independent variable. Okay, so now planning for scientific investigation, just quickly and shortly, I know you guys are tired. Ethical issues, meaning you have to have permission. You must ask, may I take photos? May I have an interview? Logistical arrangements. If I'm going to look or test somebody's blood for certain substances, I need to know, I need to have somebody that can draw the blood, and there's certain procedures, except for the fact that I need their permission. I must make arrangements, logistical. Where am I going to do it? They most probably will do it privately. The person must be, specialized. The person must know how to do this. And then how am I going to do this? What is the method, the procedure? And when I have all the data, how am I going to analyze and present and record the data? Those 4 things, very important. Okay. Ladies and gents, just a reminder. Now, just a few questions to look at. You can actually stop the video now and go and take a breather, and then just come back and work through this with me, because it doesn't help when you have the knowledge but you don't know how to apply it. I've seen many times you guys and girls actually know the work, but when they ask the question, you are so confused. You don't know if you are author or Martha. Okay, so here we start. Section A, question 1, multiple choice. Okay, let's just do 1 or 2 of the multiple choice. Let's do this one, 1.1.2. Which one of the following shows the correct path of air movement during exhalation? Now, there's the trick. Don't now think about inhalation, exhalation, when you breathe out. Okay, when you breathe out, we start at the alveolus. So there I have alveolus, there I have alveolus. So immediately I know not relevant, I can scratch them out. Now I have a 50% chance of getting the mark. So I start with the alveolus, and then it's bronchiole, then bronchus, and then trachea. So they just check if you know the difference. This is the big one, this is the little one, so it's littlest one first and then the rest. So the answer is B there. Okay, so we look at all of them, very important, 2 marks each, 10 marks for that section. All right, now we go to the terminology. Again, let's do this one, 1.2.2. The tube that connects the urinary bladder with the exterior. So kidney and bladder. So it works like this. There's the kidneys, there's the bladder, there's the ureter, this is the urethra. They say the tube that connects the bladder with the exterior, so it's this one. Your answer is urethra. Okay, fit column A to B or column 1 to 2, you can only use A only, B only, both A and B or none. So let's do this one, 1.3.2. Keeps the trachea and bronchi open. It will not be the cilia, it will be the C-shaped rings, which is B only. All right, now let's go to the longer questions, and there you have it. They give you a picture of the nephron, which is the functional unit of the kidney, and now they're going to ask you questions. Let's see, what's the first one? Identify the above structure, and that would be the nephron. Okay, this is the nephron. Name the 2 main parts of the structure. We have this part and this part. Okay, this is called the Malpighian body. So we have the glomerulus and Bowman's capsule, and the 2 together is called the Malpighian body. Then this whole section is the renal tubules, which is made up out of the proximal convoluted tubuli, loop of Henle, distal convoluted tubuli, and the collecting duct. But they are asking the main parts. So the main part is, it's about the kidney, so it's the renal tubule, tubule, okay? 2.1.3, name the process that occurs at the part labeled 1. What is happening there? Filtration is happening there, or you can say glomerular filtration. Okay? What is the process happening at number 3? There's number 3, so that will be reabsorption. It happens in the tubes, so it's tubular reabsorption. And that is how it gets more intense as we go along. Now they say 2.1.4, provide labels for vessel A and B. Now I know their names are almost the same, but I told you the one that comes first is afferent and the one that is second is efferent, because A is first in the alphabet. So vessel A will be the afferent arteriole, vessel B will be the efferent arteriole. And we can also ask questions about why this one is wider than that one, etc. 2.1.5, explain the role of A and B in the process named at question 2.1.3a. So glomerular filtration, and this is exactly what I talked about. So afferent, the afferent arteriole will be wider, and the efferent arteriole will be narrow, because this one is supposed to force blood under pressure so that filtration can take place. And filtration of what? That will be filtration of the substances that is small enough to go through. It's 3 marks, so that should cover that. The afferent arteriole is wider than the efferent arteriole. Why is that? Because it needs to put blood under pressure or great pressure so that the substances can be forced into the space so that filtration can take place. And this one, there it is, homeostasis. I told you, homeostasis of what? Sodium, salt. Okay, so here you can do the mind map for yourself. You can do that mind map for yourself, but you must describe, so you must use the words. So first of all, you will say, when the sodium levels, and I'm just going to do the shortcut, when the sodium levels are high, what is secreted? Aldosterone for salt, the hormone aldosterone, and I have to secrete less, so aldosterone secretion is decreased. When this is decreased, there will be less sodium, which is the salt, reabsorbed. And where is it reabsorbed? That will happen in the blood capillaries. So more sodium is now excreted through the urine outside of the body, and then the amount of sodium in the blood is decreased back to normal. So please just check your mind map, negative feedback for sodium, for water, and for CO2, because your teachers can ask you that. All right, let's see what else do we have. Okay, inhalation, exhalation, and there they say, tabulate 2 visible differences between the 2 stages. Things that you can see here. So things that I can see is air is going in, air is going out. There's the diaphragm shape. I can see size of thoracic cavity decrease or increase. So it's very important. It must be a table. Obviously, you will get a mark for the table, and then I guess it will be 2 times 2. 2 things here, 2 things on this side, 2 things on that side. But the important thing is it must be visible. So you can say for A and B, you can even say for yourself, this is inhalation, that is exhalation. So here you can say air is moving in, air is moving out. You can talk about the diaphragm. In A, what is the diaphragm doing? It's moving down, it's contracted, it's flattened. What is it doing in B? B, it's more arched, it's relaxed. What else can I see? The volume of the thoracic cavity increases. The volume of the thoracic cavity decreases here. The rib cage is moving up. The rib cage is moving down. So remember, look at the question. Don't let them trick you. Okay, I almost finished, ladies and gents. Here we have those graphs about the curves. They say a group of learners wants to investigate. The moment I see this, to investigate, in case they ask me dependent, independent variable, in case they ask me investigative question or aim, or to do the hypothesis. All right. 3.2.1, provide the graph with a suitable heading. Obviously, then I go back to that. Okay. To investigate, what am I investigating? The change in population size of 5 different species over time, or the change in population size over time, or the change in population size of different species over time or with relative age. All right. Now identify dependent variable. Dependent variable is there, number of survivors. Independent variable is there, relative age, or you can say time. And then, this is now what you have to study, part of your population studies there. Which curve A, B, C, D, or E, there's the different lines, the curves, indicates that the same percentage of young and old individuals die. You can see it's number C, it's proportional, okay? As you get older, it's the same. So that is C. Mortality, that is death, remember. Death is concentrated in older animals, and the possibility of death increases with age. Which one will that be? That will be number A. And why is it looking like that? It looks like that because in the beginning, the parents, it's looking after the young that is inexperienced and not protected. They can die if there's no parental care, but here's parental care. But now the parents, they get older over time, and then their numbers decrease because they die. Okay. Then 3.2.4 indicate whether the following factors will increase or decrease the population size. Immigration, meaning more organisms coming in, that is going to increase the numbers. Natality, the natal wing of the hospital, that is the babies, so it's birth, it's going to increase the numbers. Okay, just to see if you understand the terms. Last one. We have a case study, and this case study is all about the elephants, the culling, all right? Now, very important, ladies and gents, when you get a case study like this, yes, some of you are going to complain because you don't like reading, but you're always lucky. It's like a lucky packet. There's always somewhere a question that you can get directly from this passage. So just do yourself and the teachers a favor and read this very closely. So the first one is what is meant by culling. We talked about that. That's the legal killing of animals to reduce the effect or the negative effect that they can have on the environment. Then they say, what evidence from the above passage? That's like Christmas. So you must get the information for this in the section. You have to go and read it. Now, scientists are blaming humans for the elephants' damage to trees throughout the park. So this is what they are saying. Now you have to read through this whole thing, okay, to see. For instance, quickly I can see there, the park's scientists suggested that culling should not take place. The following points were discussed by the scientists before a decision about culling was made. So they talk about the scientists. If elephant numbers are allowed to increase to carrying capacity, limited food and other factors will eventually slow down the growth rate of the elephant population. Okay, so they talk about what you need to do. And then they say here the presence of artificial watering points throughout the park. So in other words, not the natural ones, not the dams or the ponds or the rivers or whatever. They put watering points there, like when we don't have waters and they bring the tanks. So you bring watering points to try and make the animals disperse over an area, that they do not all concentrate in one area. So they say scientists are blaming humans for this, because if the humans did this, the animals will not now concentrate in a specific area. So they are blaming the humans, and there's our reason. So you just give that passage as proof. Then elephants are not the only threat to the reduction of biodiversity. And here they say the impact of global warming suggests that many plant and animal species found in the Kruger Park will not survive there within a few decades. The culling of elephants to save them is therefore ineffective. Elephants are not the only threat. So they are saying that global warming is also a threat, and that is your information. So ladies and gents, there you have it. Just quickly through exam or formal test question paper, you have to practice. And please, I'm asking again, read your information properly so that you know what is happening. Okay, if I can just have you ladies and gents back with me here. Thank you for being patient, and please grade 11s, I know the pressure is on, you know grade 12 is getting closer, your marks are very important, but the first thing is make sure you know the theory, and then you have to find time to practice 1 or 2 papers and questions, because like I said, I've seen many times, you know the work, but the application, you don't understand the question. And the only way you're going to be able to understand this question is by practicing. So good luck, and I hope it goes well with all of you.